Transgenesis is the process of transferring a gene known as a transgene from one organism into the genome of another. The resulting transgenic organism will express the gene implanted into its genome and exhibit new internal or external characteristics. Transgenesis has three main steps. Firstly, gene chips are used to identify a target gene or protein that codes for the desired trait. They do this using DNA probes manufactured to stick to the target gene. Secondly, the target gene is isolated by cutting the DNA with restriction enzymes into fragments. These fragments are separated according to size using gel electrophoresis. The target gene is cut from the gel and amplified using PCR or bacterial plasmids, otherwise known as gene cloning. After isolation, a viral vector is used to transfer the target gene into the nucleus of the cell in the organism being modified. A reporter gene is often incorporated to allow researchers to identify which cells have integrated and are expressing the transgene. A reporter gene codes for an easily observable and selectable characteristic, for example, glow-in-the-dark protein in transgenic mice. Prompter and termination sequences are also incorporated either side of the transgene to control the rate at which genes are transcribed and, as a result, control net protein expression and increase likelihood of expression. Current uses of transgenics include Bt cotton, which is a genetically modified cotton variant engineered to produce a toxin called Bacillus thuringiensis. This toxin is harmful to lepidopteran larvae, pests that damage the cotton crops. Insertion of the gene causes the cotton plant cells to produce crystal insecticidal proteins or cryoproteins that kill the worms. Transgenic mice have their gene sequence altered through introduction of a foreign DNA transgene into their genome. Transgenic mice are used to understand the way genes function, to inform future biotechnologies, and importantly, are used to understand human disease and determine possible treatments. Future uses of this technology include designer babies. Designer babies are those whose genetic makeup has been altered to ensure selected traits or characteristics. Using transgenics, scientists could delete or edit genes that code for genetic diseases such as haemophilia. It is possible that through generational selection, genetic diseases could be eliminated from the human genome entirely. In the same way, selection could target physical traits such as intelligence or eye colour. Personalised medication improves the administration of drug therapy by tailoring it to the needs of the individual using genetic epigenetic information and/or a profile. Doctors can test cancer cells with certain genes and protein changes to determine the best targeted therapies in the treatment. Some advantages of transgenics in agriculture include increased crop resistance to pests, disease, and drought, which increases crop yields, enabling crops to be grown in varying harsh climates. The reduced use of pesticides could also improve human health. Altering crop quality could also increase levels of protein and overall nutrition, which could combat common human illness and improve overall public health. Together, farming industries and associated communities worldwide also benefit, due to increased annual profits and greater workforce opportunities. In medicine, xenotransplantation, the process of implanting an organ from an animal, often pigs, to humans, we could design the animal's organs without the gene responsible for the human rapid immune rejection response, making organ transplants more effective and more available worldwide. However, there are disadvantages to transgenics. These include environmental risks. There is a risk that transgenic organisms may have unintended consequences on ecosystems, such as harming non-target species, disrupting food chains, and introducing new diseases or pests. Due to transgenics being a new technology, there is also little information about its long-term effects and impacts on human health and the environment. For example, Bt cotton's toxic leaves could affect animals who aren't pests. Designer babies pose an ethical challenge in transgenics. This technology could be taken advantage of through the selection of appearance instead of the removal of genetic diseases. The selection of common physical traits such as blue eyes and blonde hair has the potential to reduce human genetic diversity and/or amplify the possibility of mutations due to inbreeding. In conclusion, when considering the benefits of transgenic crops compared to the risks of designer babies, I believe that with management of misuse in the technology in the form of designer babies, as well as conservation of ethical practices and environmental impact, the use of transgenics could be very beneficial to our society. Finally, the portrayal of transgenics in the media can often be misleading. An example of this is Jurassic Park, a sci-fi action film about an island theme park populated by genetically engineered dinosaurs. Despite this movie's popularity, the science behind its concept is inaccurate. In the film, Mr. DNA explains that DNA extracted from a mosquito was 80 million years old. DNA only has a half-life of 521 years, meaning that in a maximum of 6.8 years, all the DNA would have broken down or ceased to exist. The oldest DNA extracted from the molar of a mammoth was only 1.2 million years old. meaning that the fossilised mosquito couldn't have contained any living DNA. The choice of using frog DNA to patch the dinosaur's genome in the film was used to develop the plot. The possibility of combining frog DNA with foreign DNA is possible today, for example, transgenic mice, which are used to study human disease. However, if the choice were made today, a closer linked species to the dinosaur could be used. For example, a crocodile would make the dinosaur hybrid genome more closely related. Considering the inaccuracies of DNA's lifespan and the use of frog DNA, it is clear that Jurassic Park is meant to entertain its viewers and is an inaccurate portrayal of transgenics.